Hi, Miss Nikki here. Welcome to Chapter 7. We're going to talk all about cellular respiration. Basically, if we just broke it down to the simplest form, we're taking the energy out of the bonds of glucose and we're storing that energy in the bonds of ATP. Cellular respiration is the release of energy from glucose, and we use this energy to synthesize ATP. This ATP molecule can go anywhere in the body. We can break the phosphate off and we can release energy to do work. So remember, we're talking about all muscle activity requires ATP. So that's skeletal, cardiac, smooth, all neuron activity. So for one neuron to talk to another neuron, that requires ATP. So ATP is really critical, and so is this entire process, to the point that if cellular respiration is not occurring, you die, right? So this is really important. Here we can see the equation. Glucose, so this is the food that you eat, and oxygen, the air that you breathe, is used. We take the energy out, store it as ATP, and the waste product are carbon dioxide that we exhale, and then water. And remember, the majority of our body is water, but if we have excess water, we can also eliminate that via the kidneys. Before we talk about the 4 steps of cellular respiration, we have to talk about these coenzymes, NAD plus and FAD. And they're going to pick up electrons, and they're going to carry those electrons to the electron transport chain. This is the 4th step in the process of cellular respiration. And here are the long names. I just call them NAD plus and FAD. So they're going to pick up electrons and some hydrogens that we'll see as we walk through the steps of cellular respiration. And these ultimately come from your diet. Kind of the precursors for both of these coenzymes come from vitamins. So this is B3 and this is vitamin B2. Here they're showing you the cycle. Basically, if I have NAD plus, remember that plus means that this molecule is slightly positive, right? That means it's missing some electrons that it would normally have. So NAD plus can pick up hydrogens and electrons, and it forms this NADH structure that we're going to talk about quite a bit. And then NADH can drop off electrons and hydrogens and come back to its initial coenzyme form of NAD+. So basically this molecule is picking up electrons and hydrogens and dropping them off someplace else. Now we can look at the 4 phases of cellular respiration. So we have 1 glycolysis, 2 preparatory, 3 citric acid cycle, and 4 electron transport chain. So all of these are going to generate those NADHs that we talked about, and they're all going to get funneled to step 4, electron transport chain. You may have heard of this called the Krebs cycle. We're trying to stay away from eponyms named after people, so now we call it the citric acid cycle. Here's a nice overview with a figure for cellular respiration. So here's glycolysis, number 1, 2 preparatory, 3 citric acid, and 4 electron transport chain. And you can see all of the NADHs and the FADH2 that are being created during this phases 1 through 3 are all being driven to the electron transport chain. And this is where we make the majority of our ATP. What they want you to take away from this image is, first, that both animal cells and plant cells do respiration. Both of these have mitochondria, right? So both plants and animals are able to do cellular respiration. The second takeaway is where these occur. So glycolysis, which is step 1, occurs in the cytoplasm. The preparatory reaction is kind of the link between these two, and you'll see more about that when I get there. But these last 2, stages 3 and 4, these occur inside the mitochondrion. First, we have glycolysis. So we're looking at step 1, stage 1, whatever you want to call it. And we're going to take glucose, which is a 6-carbon, and we're going to break it down to pyruvate, which is a 3-carbon, and we're going to have 2 of those. We're going to create a little NADH, and we're going to create some ATP, just 2. They break glycolysis down into energy investment step and energy harvesting step. So what they're saying is you have to put energy in to start glycolysis. So you see here we're adding ATP. We have a 6-carbon, we're adding some ATP, We're phosphorylating the molecule, and this causes a split. So now we have a 3-carbon molecule here and a 3-carbon molecule here. So we added energy and we added a phosphate, and that caused the 6-carbon to split into 2 3-carbons, and we call this structure G3P. Now we move to the energy harvesting steps. Remember, we had G3P here, and it's a 3-carbon structure. NAD coenzyme is going to come in. Remember, it's going to strip off electrons and hydrogens. So we're kind of modifying this molecule, this 3-carbon molecule. We're stripping off electrons and hydrogens. We add a phosphate. So now we have this molecule, which is called BPG. And you can see it now has 2 phosphates. Up here with G3P, it just had 1 phosphate. Now ADP is going to come in. ADP is going to form ATP. What do you need to form ATP if you're ADP? You need a phosphate, and you need to break this bond and get energy out. So ADP comes in, it pulls off energy and a phosphate and becomes ATP. So you see how the phosphate is missing? Now we have 3PG. Some water is going to come off. We're going to have a little dehydration. And now we're going to do the same exact thing again. We're going to break off this phosphate, break this bond. We're going to take energy out and the phosphate, and we're going to make more ATP. And our final product with glycolysis is pyruvate. And you can see this is still a 3-carbon. So what have we done this entire time? We've stripped off electrons and hydrogens. We added phosphates, and then we removed phosphates, and we also removed energy. So basically, we've been manipulating this G3P structure to pull off electrons, hydrogens, and energy, and we end up with this pyruvate structure. They're still 3-carbon structures, it's just been modified. So they like to talk over here about the amount of ATP that's being generated. So in this stage, we generated 2 ATPs. And then here in this stage, we generated 2 ATPs. That's supposed to be a P. So we created 4 ATPs. That's the energy harvesting step. But go back. Previous slide, we had the energy investment. Remember, we were at a -2 ATPs. We added 2 to start this. So if I make 4 and I had to use 2 to start it, what's my net? How many ATPs did I actually gain? This is kind of like your paycheck, right? This is how much you get paid, and then we take out taxes and you end up with your net pay. Here's just an example of how this ATP is created. There is an enzyme responsible for it, so you'll hear it called sometimes substrate-level ATP synthesis. And so you're probably going to most closely identify this with glycolysis. So if you hear substrate-level ATP synthesis, this is happening in glycolysis. And you can see we have our structure here, our BPG, and we're going to have the phosphate fit into the active side of the enzyme. We have ADP fitting into the active side of the enzyme, and it's going to bring these molecules close together, break this bond, release energy, and now you have your ATP structure. I recommend that you do this for all 4 phases. Do an input and an output. So you can see here inputs. What did you put in? You put in a 6-carbon glucose, and you broke it down into 2 3-carbon pyruvates. You put in 2 NAD pluses, and you got out 2 NADHs. Remember, that's where we stripped the electrons and the hydrogens. You put in 2 ATPs, and you phosphorylated the molecule, so you ended up with 2 ADPs. And then we added 4 ADPs plus 4 phosphates, and we got a total of 4 ATP out. Again, the net gain is 2. We put 2 in, we got 4 out, so our net is 2. We just saw glycolysis, and we know that glycolysis is when we take glucose and we turn it into 2 pyruvate. sometimes fermentation occurs. So instead of pyruvate, you can create lactate. If you're an animal, lactate. If you're a plant, alcohol and CO2, carbon dioxide. So sometimes, let's say if you have low oxygen, so you're in anaerobic respiration, you're going to create lactic acid. So we haven't gotten to the other 3 stages of cellular respiration, but remember, cellular respiration has 1, which is glycolysis, 2, which is the prep, 3, which is the citric acid cycle, and 4, which is the electron transport chain. In the electron transport chain, oxygen is critical. So if you have low oxygen, there's no point in doing these 3 steps because you have to have oxygen at the end in phase 4. So if you do all 4 phases, we usually call this aerobic respiration. If you have low oxygen, we call it anaerobic respiration. And if you have low oxygen, instead of doing these 3, we basically just have this glycolysis, but creating lactic acid instead of creating pyruvates. I mention it because lactic acid is one of the chemicals or items that makes you feel sore after you work out. It's usually cleared out of your system within a couple of hours to half of a day. But this is why athletes want to stay in aerobic. So think of aerobic as, you're running and you're breathing deeply and you're getting enough oxygen and everything's fine. If you're running and then all of a sudden you're doing that, like you're really trying to take deep breaths, you probably have low oxygen, you're probably in anaerobic respiration, and you're making lactic acid. If you're a performance athlete, you want to stay in aerobic. It's the most efficient. This, you're still making 2 ATPs. You're just kind of biding time, right? You're not getting to this last step where oxygen is critical, where we make the majority of our ATP. So it's kind of like a holding pattern. But it's good for plants, right? How would we create wine or beer if we didn't have fermentation? Here's a nice figure. They didn't draw both sides happening. They just put a 2 in front of everything. But you can see it's almost the same. We still have G3P. We still have BPG. We're still stripping off electrons and hydrogens. We're still creating ATP. We get to this pyruvate, and instead of taking pyruvate and moving it into the preparatory reaction, instead it gets broken down into lactate in animals and bacteria, or alcohol in plants and yeast, and one of the byproducts is also CO2. So this is why when you take yeast and you make bread, the CO2 that comes off from fermentation is what makes your bread have little holes or pockets in it, right? Light, fluffy bread. So fermentation is useful. We've exploited it in the plant and yeast population to make yummy food for us or drinks for us. In animals, it's really just biding your time until you can breathe normally again and get enough oxygen to push the pyruvate through the last 3 stages of aerobic respiration. So if we're doing aerobic respiration, we take this pyruvate and we're going to tag it with something called CoA. And this CoA is needed for this molecule to enter the mitochondria. So that's why they call it the preparatory. You're prepping this molecule to enter into the mitochondria and finish phases 3 and 4. So the pyruvate, this is a 3-carbon, and there's 2 of them. They didn't show you both. They just put a 2 in front of everything. And this pyruvate, we're going to have the coenzyme come in again and steal more electrons and more hydrogens off of the pyruvate. We're going to add this CoA tag. So now we have an acetyl group or an acetyl CoA. So this is a 2-carbon with a CoA attached, and the other carbon comes off as carbon dioxide. So what did we do during the prep? We stole electrons and hydrogens off of the pyruvate. We added the CoA to the pyruvate, and we got carbon dioxide as a byproduct. Now we move to the citric acid cycle. So I like this image. It kind of shows you what we've already covered, and we can see that that preparatory reaction is needed to put that CoA tag so that we can get inside of the mitochondria. So now we're going to look at the citric acid cycle. I know it looks like there's a lot going on here, and there is, but let's just walk through it. I'll probably walk through it multiple times in different aspects for you rather than trying to write all on this one slide. So here's the citric acid cycle. We just came out of the preparatory reaction. We have our acetyl-CoA. So we've got a 2-carbon group with this CoA added. And you can see that that's the ticket to get into the citric acid. The CoA comes off and the 2-carbon enters the citric acid cycle. So I've got my 2-carbon here entering the cycle. The starting molecule is a 4-carbon. So if I take a 4-carbon and I add it to a 2-carbon, I end up with a 6-carbon, the citrate. Then what's going to happen? The NAD coenzyme is going to come in. It's going to strip off electrons and hydrogens. We're also going to strip off one of the carbons, and it comes out as carbon dioxide. So we went from a 6-carbon, we pulled off 1 carbon, so now we have 5 carbons. Guess what happens? NAD plus comes in again. It steals electrons and hydrogens, and we have another carbon coming off in the form of carbon dioxide. So now we go from a 5-carbon to a 4-carbon. We're also going to generate some ATP. So what did we have to steal off of the molecule? Some energy, right? And then we have finally our FAD. So this is the first time that we've seen this FAD coenzyme. It's going to come in and steal electrons and hydrogens, and we still have a 4-carbon molecule here. We're going to steal some more electrons and some more hydrogens, and now we're going to go back to our original 4-carbon structure so that when the next acetyl-CoA comes in, it has something to bind to. So here, let's just look real quick. We know we're generating 1, 2, 3 NADHs. We're generating 1 ATP, and we're generating 1 FADH2. This was from 1 molecule from the preparatory reaction. But remember, we had how many? We had 2 pyruvate come in, and those 2 pyruvate were modified into 2 acetyl-CoAs. So this citric acid cycle has to turn twice in order for the 2 pyruvates from 1 glucose to be processed. So we have 1 glucose molecule. How many times does the citric acid cycle have to turn to process 1 glucose? So times 2 turns of the citric acid cycle. So all of our products we have to multiply by 2. Here we have our nice input and output charts, but remember we're saying 1 glucose, and during glycolysis we turned it into 2 pyruvates, and then the 2 pyruvates we turned into 2 acetyl-CoAs, right? So 2 acetyl-CoAs went into the citric acid cycle, and we got out 4 carbon dioxides. So something to note, the glucose carbons, so there, I don't want to draw them all, but I guess I'm forced to, our 6-carbon glucose structure. If I tagged these carbons radioactively with dye, they would come off as the carbon dioxide. Also remember here we made 2 acetyl-CoAs, we also made 2 carbon dioxides. So these guys would come off as tagged, and all these carbon dioxides. So what I'm trying to say is the carbon dioxide that you exhale, those carbons ultimately came from glucose. We know that the 2 coenzymes came in, they stripped electrons and hydrogens, and we generated a lot of NADH and FADH2. And we know that each turn gave us 1 ATP, and that this has to turn twice, so we know we have 2 ATPs total from 1 glucose molecule. So you can also look at this image and tell me the outputs with 1 turn, right? If we just did one turn, we'd have 2 carbon dioxides, 3 NADHs, 1 FADH, and 1 ATP. If we're talking about outputs per glucose molecule, then you're going to use these numbers. 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs. Here we have our image of the electron transport chain. We've covered these 3 phases. You can see that glycolysis, preparatory, and citric acid, all 3 phases made NADH, and FADH2 was made just in the citric acid cycle, but that's all getting funneled into the electron transport chain. We also can see that not so much ATP has been made so far, right? We've only made 4 ATP. Preparatory reaction, no ATP made at that stage. The majority of our ATP is going to be made here at the electron transport chain. Just to make sure that we understand what we're looking at on the next image, here's our mitochondria. Remember, it has 2 layers, right? It's a double membraned organelle. The space inside is the matrix. There's also a space between the 2 membranes, and that's called the intermembrane space. And there's these infoldings of the inner membrane, and they're called cristae. So we're going to look at a close-up. It's kind of like we just blew up this one section, this cristae. So there's going to be proteins embedded here in this membrane layer. Here's the cristae, right, the infoldings, and then this is the matrix. So remember what happens. All of the NADHs and all of the FADH2s are going to come in and drop off electrons into this system. So this here is the electron transport chain. It's basically enzymes, and enzymes are proteins, right? And these complexes that the electron is going to move through. So NADH releases its electrons that it was holding, and it releases some hydrogens. The electron travels through the electron transport chain, and the final acceptor for that electron is oxygen. That's why you have to have oxygen to complete this step of cellular respiration. If you didn't have oxygen, this system would back up and it would stop working. So NADH and FADH2 drop off the electrons move through the complex, and the final acceptor of the electron is oxygen and hydrogens to form water. Remember, when NADH dropped off electrons, it was also carrying hydrogens. The hydrogens move through these enzymes or proteins as the electron moves through. So it's kind of like as the electron gets here, hydrogens are pushed from one side of the membrane to another. And you can see that happening repeatedly. The hydrogens build up out here. So you have a low hydrogen atmosphere inside, and you have this high hydrogen atmosphere outside. Let's follow the hydrogens. So we know that the NADH and the FADH drop off electrons, but they also drop off these hydrogens that are being pumped through the electron transport chain. The hydrogens are going to build up. Remember, I said they're high in concentration on the outside. So these hydrogens are going to build up, come to the other side where there's an ATP synthase complex. So what's going to happen is the hydrogens are going to flow through this enzyme, and as the hydrogens flow through, they kind of whip this enzyme around, and it generates energy. So as the hydrogens flow through, they generate energy, and then ADP plus P takes that energy and forms ATP. And then the ATP is shuttled outside of the inner membrane. So they call this process chemiosmosis. So chemiosmosis is really creating ATP using a hydrogen concentration gradient. So what's the only reason we're making ATP? We have a protein, we have hydrogens that are high on one side and low on the other. We're taking those hydrogens, they're being funneled through this enzyme, and the force or the energy from those hydrogens flowing is what creates the ATP. So if you hear the word chemiosmosis, think ATP creation based on a hydrogen concentration gradient. Let's follow this one more time. So the NADH and the FADH are going to drop off electrons and hydrogens. As the electron moves through the electron transport chain, hydrogens are going to be pushed through the molecules, the proteins, the enzymes. Finally, the electron gets to the end of the electron transport chain. Oxygen and hydrogens combine to form water. So the electrons that we stripped off of the glucose in various steps is going to end up in water. The hydrogens get pumped through. The hydrogens come back to this side, and they're going to go through the ATP synthase complex, and the energy from that hydrogen ion flowing through that enzyme is what allows ADP plus P to become ATP. And then that ATP is shuttled out. This is a nice representation from the textbook showing you the location and then the products for each reaction. So here we have glycolysis. Glycolysis happens in the cytoplasm. We take glucose, a 6-carbon, and we break it down into 2 3-carbon pyruvates. We generate 2 net ATPs and 2 NADHs. Then we have our preparatory reaction where we take the 3-carbon pyruvate and we break it down or turn it into a 2-carbon with a CoA attached, and then one of the carbons comes off as carbon dioxide. But remember, we have 2 pyruvates, so we have to do everything twice, right? So then the 2 2-carbon CoAs enter the citric acid cycle. They're going to generate 2 ATPs, 4 carbon dioxides, 2 NADHs, 2 FADHs. And then all of these are going to get funneled into the electron transport chain, and this is where the majority of our ATP is generated. Remember that oxygen has to be the final electron acceptor from the electron transport chain, and that we're also going to create water. So this should kind of put into perspective, we start with glucose and oxygen, and we yield CO2 and water and ATP. So we add the glucose, we add the oxygen, we end up with CO2, water, and lots of ATP. So this is where we get anywhere from 36 to 38 ATP molecules from one glucose molecule. What I would do is go through this figure and talk about glycolysis and list all the inputs and all the outputs. We kind of already went over it. I'm just telling you this is what I would do to help myself learn the material if I was doing it for the first time. I would go through the preparatory reaction and list all the inputs and outputs, all the inputs and outputs of the citric acid cycle, and then all the inputs and outputs of the electron transport chain. If you can list these out from memory, not using your notes, then you probably have a really good understanding of cellular respiration. So I'll talk to you next video. Bye.